All right, so without further ado, I will kick off our seminar today by introducing our speaker for today, Dr. Dave Block. We, like I said, have been lucky to be good friends with Dave from kind of numerous years ago. Today, Dave will be talking about the development of plant-based and inexpensive cell culture media to power the cultivated meat field. As many of us know, cell culture media is projected to be 99% of the marginal cost of the product for cultivated meat. So when we talk about some of the key hurdles to building a scalable food system with these novel food technologies, will become increasingly important to lower the cost of that cell culture media, to optimize it for food production rather than utilizing kind of pharmaceutical grade ingredients as borrowed from the biomedical field. And Dave will talk about a lot of the really exciting research that they have underway at UC Davis, one of which is generating a viable alternative to this expensive pharmaceutical grade cell culture medium by utilizing a really multidisciplinary approach and allowing us to optimize cell culture media for cultivated meat production. Dave is the department chair in viticulture and enology at the University of California, Davis. He holds the Ernest Gallo Endowed Chair. And since joining UC Davis, he's conducted research on a lot of various topics, from fermentation optimization methods to metabolic engineering of yeast for improved wine production, as well as single plant resolution, irrigation sensing and control. So most recently, his research has focused on computational fluid dynamic models for red wine fermentation and process optimization for cultivated meat production. We are, again, really thrilled to have Dave as a GFI grantee and cannot be more excited that he's joining us today for this seminar. So Dave, whenever you're ready, please feel free to take it away.
Great. Thanks, Amy. I'll share my screen. And great. Well, thank you very much for inviting me. It's great to be here. Good morning, good afternoon, good evening. I know people are all over the world, so thank you for joining and listening. And again, thank you to Amy and the Good Food Institute for the invitation to speak and for the funding for some of the research that you're going to see today. So what I'd like to do today is, first of all, do some acknowledgments, start off right off the bat. I'm going to talk about some of the work that we're doing here today. And when I say we, I really mean the two students that we have working on this project so far, Zach Cosenza and Ted O'Neill, my collaborator, Keith Barr at UC Davis, and my colleagues in the UC Davis Cultivated Meat Consortium. And of course, I'd be remiss if I didn't acknowledge all the funding that we have for this area, including the Good Food Institute, New Harvest, and NSF. And I'd also like to thank, you'll see as I go through how we got into this area, I'd also like to thank all of my colleagues at GFI, Liz, Elliot, and Amy, and others who helped us along the way to get to where we are now with our project. So I'm going to start, because people are all over the world and may or may not be familiar with UC Davis. I'm going to start with just a brief introduction to UC Davis, and I'll talk a little bit about further about my background as well. So University of California at Davis is one of the 10 campuses of the University of California. We're located in Northern California, probably about an hour to an hour and a half from San Francisco, about an hour from Berkeley. Davis is, and I apologize because this is a little bit of an old slide, but I'll point out which things are still relevant, which things are not. But some of the things that are important, it's a fairly large campus. We're up to about 39,000 students at this point. I'd say some of the things that are a little bit different from this slide, which is a few years old, we're up to about $900 million in research funding annually. Um, During the pandemic, we don't have 20,000 bikes on campus. It does make it interesting to walk around campus with all those bicycles. Um, we are first in the nation in the U.S. for agriculture. We're generally one or two in the world, depending on the year, with some of the schools around the world, like Wageningen and Cornell and so forth. And we're 10th in the nation in the U.S. for public universities. So it's a very large land grant university that's used to dealing with agriculture and food and things like that, which is really important. Just a few words on my background, just to add to what Amy said. My area is chemical engineering. My field is chemical engineering. I've been doing fermentation process development now for over 30 years. Initially, before I joined UC Davis, I worked in the pharmaceutical industry doing fermentation process development. And for those of you coming from companies, you'll identify with this. A lot of what I did in the 5 years I worked in the industry was media and process development and scale up. I did fermentation process development from 100 milliliter scale up to 160,000 liter scale, 40,000 gallon scale. Um, and so I got lots of great experience with all kinds of organisms, mostly microbial. But in the last few years I was at Roche, I also worked on building new manufacturing facilities. And those facilities that I worked on building were all cell culture facilities to produce monoclonal antibodies. And so I've had experience relevant to a lot of what we're going to talk about today. Um, So this is kind of the outline for what I'm going to talk about today. First, I'm going to give some context. Not that most of the audience here needs that context for why cultivated meat is important, but I'll kind of get everybody up to speed on that first. And then I'm going to talk about the UC Davis Cultivated Meat Consortium that's been developing over the last couple of years, and talk about what we're doing there, because it's kind of a unique organization. Um, and I think a developing organization that's worth talking about, so that people know what's going on uh, more broadly than what I'm doing in my lab, and can connect to it if there seems to be a reason to connect to it. Then I'm going to talk a little bit about the research that we're doing, and I have to say, we're as we've been working in this area for almost exactly 2 years, which is not a lot of time. And so I'm going to talk about where we're starting and the directions we're going, and then there'll be plenty of time afterwards to talk about questions and have a discussion about this area of optimizing media for cultivated meat production. So that's kind of the game plan. And so let's start with the context. The context is we're focusing on cultivated meat. I will say, in the past, I've worked on protein alternatives produced through fermentation and so forth. But today, what we're going to focus on is cultivated meat. So obviously, in conventional animal agriculture, you're growing an animal and you're getting the meat out of that animal, whether it's beef or chicken or pork or seafood or whatever it is. And what we're talking about here, we're focusing on today, is this idea of going from the animal, isolating cells from the animal, growing them basically in a fermenter or multiple fermenters, and somehow getting either unstructured product like a hamburger or a structured product like a steak out of um, out of the process. And so that's what we're going to focus on today. So why is this important? I think a lot of people on this call understand why this is important. It's important because the population of the earth is growing tremendously, and the production of food is not keeping up with that. And so this is on the left information from the World Food Center here at UC Davis. They're estimating a need for 60% more food by 2050 to feed the growing population. And so when you think about that, and obviously an important part of that food supply is going to be dietary protein sources, there aren't that many solutions available or types of solutions, and I have them characterized here on the right. And one of them is to expand conventional animal agriculture. And I think there are all kinds of potential issues with that, from land use issues and food safety issues and um, animal welfare issues and things like that. Although it's likely to remain an important source of dietary protein for the foreseeable future. We could develop protein alternatives. So these be plant-based or microbe-based alternatives, and that's going to be, it's already a viable source of dietary protein, but that will, I'm sure, continue to expand. And the other one that I'm going to focus on here is developing new means for producing meat through cultivated meat, through growing cells in a fermentation process to produce actual meat, but in an alternative way from conventional animal agriculture. So that's the one. The third one is the one we're going to focus on here, and of course, GFI is also supporting um, some of these other efforts as well. So what I want to point out here is that this is a new and growing industry. If you go back 5 or 6 years ago, there are very few companies that are around, maybe just a couple or handful of companies. Whereas in 2020, there are over 50 companies in this space. And if you look at the funding raised by cultivated meat companies here on the right, you can see almost an exponential increase. And I don't have data for all of 2020, but I found this data on GFI's website for the first quarter of 2020. And you can see even without the rest of the other 3 quarters of 2020, we're still in an exponential growth phase, which makes it a very interesting space to be in. And as many of you probably know, with all this growth, there's now one product on the market in Singapore from Just. And so there's going to be, I think, hopefully, an exponential growth of the whole industry and getting products to market. So what's keeping that from happening? Well, obviously, some of that's happening, and there are a lot of companies that are working diligently on this. But I would say the three hurdles that are out there are, first of all, cell lines. So cell lines to that cells that will grow when we need them to grow and differentiate when we need them to differentiate, which is certainly not an easy thing. And we're running into barriers just ourselves in research for that. And I know there have been discussions in some of the GFI groups that get together to discuss GFI grantee groups that's a constant barrier. The second barrier is having inexpensive media. That's the one that we're focusing on, along with the process scale up. And there is a lot of expertise out there in process scale up. There are a lot of people like me who have done this for a living, probably some of you as well. But there's going to be particular things that need to be done because this is a food product that haven't been done in the past. And so that's the second hurdle. And then creating the structure, as Amy was talking about, the focus of the GFI call this year, creating that structure like a steak or a chicken breast or salmon fillet. That's the third of those hurdles. And so in addition to the work going on in industry, this work has to be going on in academia as well. Stuff going on in academia will be open source, and that will help the whole industry moving forward. So I want to start by talking about what we've done at UC Davis and the whole effort at UC Davis, because I think it's a good model for moving the industry forward. So I want to talk about how we founded this effort, what we're doing, and where we are now. So our effort really started about 2 years ago, or just over 2 years ago. I had a student who was talking to me about going back to some of the optimization work, the experimental optimization work that I had worked on probably about a decade ago. And we were trying to figure out what kind of system to apply it to. And I happened to be in charge of a food innovation engineering conference offered by ASCHE. And I know there's at least one or a couple people out there in the audience today that helped with the development of that conference, which is still going today. I think they're on their 4th annual conference now. And there happened to be a speaker from the Good Food Institute who started talking about cultivated meat. And I will say, a couple of years before that, I thought about it a little bit in discussions with a major food company that was looking for alternative protein sources, actually in the pet food realm, but had kind of put it aside, hadn't really thought about it until the speaker got up and started talking about it. And I thought, wow, if one of the hurdles is cultivated is media optimization, that's something that we do a lot of in my group. Maybe we can use that as a system. So went back and had several conversations with people at Good Food Institute. Um, Recruited not only that student, but another student who is looking to work in cultivated meat, and disappointed that up to that point UC Davis wasn't working in that area. That was Zach Senza and Ted O'Neill. They applied for New Harvest fellowships and got them by the middle of 2019. And then in October of 2019, all the researchers on campus, about 30 researchers on UC Davis's campus, got together to discuss starting the UC Davis Cultivated Meat Consortium. We started that in October of 2019. Um, we also got funding from the Good Food Institute to continue our work. I should say the New Harvest grants were with my collaborator, Keith Barr, and our GFI grant is with Keith, and also collaborators in chemical engineering, Karen McDonald and Soman Nandi. And that funding started about August of last year. And then almost exactly a year ago, we decided to put in a grant to NSF in their Growing Convergence program. And Elliot Schwartz was instrumental in helping us with that, along with Liz Specht. And we were awarded that NSF grant and started on October 1st. And this is looking forward a little bit, but by May of this year, so in the next couple months, we will have probably at this point now 14 or 15 faculty members on campus who are working on cultivated meat. We'll actually have probably more than 10, probably 11 or 12 graduate students and postdocs working in this area, and probably about 10 undergraduates working in this area, making it by May one of the largest academic efforts in the world focused on cultivated meat. So I want to say a little bit about this NSF grant program. It was not a program geared towards cultivated meat. In fact, what cultivated meat was not mentioned in the call at all, but it was a program that had two requirements: that the research address a very specific problem, very specific, compelling problem, um, and that there'd be deep integration across disciplines in solving that problem. And actually, when you think about it, cultivated meat falls into that category really well. And so we did apply. We did get funding. It's about $3.5 million over 5 years, with a checkpoint after 2 years. And what's I think significant about this is I believe it's the first federal funding from the United States for cultivated meat research, specifically for cultivated meat research. And so we're very proud of that and very happy to have that support. And again, thank GFI and others for helping us get through that milestone. So there are four objectives to that proposal that are here, and I'm going to summarize them. You'll see the first three of them address basically the three hurdles that we talked about before. So there's objective where we're working on cell line development, objective where we're working on inexpensive media, and focused on unstructured product. A third objective where we're looking to create structured products like steaks, for instance. And then the fourth objective is really to do a detailed techno-economic analysis and life cycle analysis to understand the technical hurdles that still keep the products from being economically feasible, and to compare the sustainability of this with conventional animal agriculture and other routes to dietary protein. And we feel that there's already some of these analyses out there, including from UC Davis, but we think that these analyses will develop as we generate more information, get more information into the public domain. And so that's the goal of that objective. I will say, in addition to just what I said, the objectives are a little bit broader than that, in that, for instance, in the second and third objectives, not only are we looking to make these products, but also to characterize their oxidative stability, which is important for meat, their nutritional value, and how that is affected by the processing, um, and also how they taste. Okay, so the sensory characteristics, including things like mouthfeel and so forth, that are going to be important to consumer acceptance. So in order to do that, we have these 13 faculty members. They come from very broad background. Um, they cover 7 departments at UC Davis and 3 colleges. Um, and you can see they're kind of color coded for area, from bioprocessing and tissue engineering through the nutrition side of things, the stem cell side of things, muscle physiology, um, and so forth. Um, and so uh, we have all these people from different backgrounds all gathered around the 4 objectives. And so for each of these 4 objectives, we have people from different backgrounds. That's going to be important to solve these problems, to have people looking at the problems from different viewpoints. Um, it's also going to be important for the students who are trained in these areas. And as part of this grant, all of our trainees will have at least 2 mentors from different disciplines. Because if we're going to solve these difficult problems, and students are going to then go out to industry and solve these difficult problems of getting product to market, they're going to have a multidisciplinary or transdisciplinary background. And so that's really important. So in addition to the research, research is obviously really important. Another important thing that I just mentioned is training, making sure that we're training the future workforce for cultivated meat industry. We will be having monthly seminars, and hopefully seminars also that are more public as well. We actually just had one last night um, with the Robert Mondavi Institute. We're looking to have, and we're already establishing interactions between industry and academia. I think that's really important, to have cross fertilization of ideas and to have interaction not only with the students we send out, but to be talking constantly about understanding what are the important problems to be solved, how are those evolving over time, what do our students need to be trained to effectively go into the industry and start quickly, and so forth. So that's really important. And we'll also be setting up at least annual extension or continuing education for the industry. These are all part of the NSF grant. Um, but I think that this is the UC Davis, being a land grant university, has a lot of experience doing this. And so one of the reasons why we can do all of this on one campus, and that's not to say we won't be collaborating with people all over the world. We're already setting those kinds of things up. But it's important because UC Davis has a history of working very closely with industries related to food and agriculture. And I put this up because my main appointment at UC Davis is in the winemaking grape growing department in viticulture and enology. And viticulture and enology has a long history of doing this. The department dates back to 1880, and is often credited with helping the industry to grow from almost nothing, let's say 60 years ago, to world class now, in 2021. Um, the department has worked very closely with the industry on research projects, on training students, and internships and continuing education. We do probably 15 to 20 continuing education programs each year for the California industry and broader. And so we know how to work with industry, and this is something that I'm very hopeful that we'll continue to do and build on with the cultivated meat industry, and helping them to grow and working hand in hand to make this a successful industry, not only for California, but broader than that. Okay, so I'm going to switch gears now and talk for about 10 or 15 minutes about what we're doing for research. And so I talked about the convergence of disciplines. This work really started with collaboration with Keith Barr, who's a muscle physiologist and works on muscle tissue culture. And my area is fermentation optimization and biochemical engineering. And what we wanted to work on was how do you develop an inexpensive plant-based growth media, or maybe a differentiation media for cultivated meat production. So, when we think about the process, and this is my simplified idea of what the process will look at. If we think about where the media is here, we're going to take a cell line and we're going to start growing it from a very small scale and start increasing the size of the scale from probably on a factor of 5 at a time, if it's anything like other cell culture processes. And when we get to the largest scale, and there's going to be other ways of doing this, but just bear with me. This is one idea. When we get to the largest scale, we're probably going to differentiate those cells into muscle, fat, and connective tissue. And if we're making unstructured product, to mix those into, let's say, something like a hamburger, okay, or ground chicken, or ground turkey, or pork, or whatever it is, ground fish. And then if we're going to make a structured product, then one possibility is we grow these cells up to a large scale, and then, for instance, 3D print them into the structure. And maybe we need media or some kind of round of growth after that to solidify the structure, affect the structure afterwards. At each of these steps, we're going to need a media, and the media may be the same for growth. It'll probably be different for differentiation. It'll probably be different for different types of differentiation into muscle, fat, or connective tissue. And it may be different as we form our structured product. So at each of these steps, we're going to need a media that's going to support cell growth of these cells. Now, when you start out in this area, I think what a lot of companies did was look to where's the experience in large-scale cell culture, and that's really in biopharma. Now, the good thing about that is that biopharma is already growing cells in cell culture, at least up to 25,000 liter scale. And I know there are companies that are doing that quite locally to us here at Davis. Within 20 minutes of us is one of the largest cell culture facilities in the world. And the question is, though, can we actually use that media in that process if the product coming out of that, which is generally monoclonal antibodies. So if you're watching TV, at least in the US, you see all these ads for new drugs that end in MAB. Those are all monoclonal antibodies produced in cell culture. And but the problem is when you go to buy those monoclonal antibodies, they cost about $1 million a pound, or because we're talking about orders of magnitude, about a million to a couple $1 million a kilogram. So obviously we're not going to be able to sell meat for that cost, and so something has to be different. And you could give up at that point and say, well, we need to come up with something different. But there is another area to look at, and that's what I show on this other side, and that's fermented foods and commodity chemicals produced through fermentation. There are lots of examples of that. And a lot of these are in the ballpark of $10 a pound or $10 a kilogram. And so that's kind of the order of magnitude that we're looking at. And so we have everything from wine and beer and soy sauce to things that are probably more relevant to what we're talking about, like citric acid. Vitamin C has one of its steps that's a fermentation step. Monosodium glutamate as a flavor enhancer, xanthan gum, which is in all kinds of food products. Antibiotics, which isn't really a food, but there's enough expertise there that the price of many antibiotics that are produced through fermentation are pretty low. And so those are actually good examples that we can look to and see what does the media look like when those food products or chemicals are produced at very large scale. And those are produced at probably easily 30,000 gallon scale or above, maybe up to hundreds of thousands of gallon scale. And so that's another place that we can look to when we're thinking about optimizing the process, optimizing the fermentation, and optimizing the media. All right. I think I'm going to start with the statement optimizing growth media is difficult. Why is it difficult? Well, this is the composition of DMEM media, which is a common, let's say, medical grade cell culture media. Um, there are a lot of components. I think there are 30 components here. I haven't counted them recently, but it's around 30 components of vitamins, salts, amino acids. One of the main components that people use with the DMEM is fetal bovine serum, which is complex, hugely expensive, and variable in quality. Um, and of course, glucose as a carbon source. There may be other possibilities. It's difficult because there are a lot of media ingredients, and a lot of these media ingredients complement each other, um, and they have synergy in the way they affect cell growth. They interact with each other. Some of them may make others less effective or more effective. They can be uh, they can supplement. Some of them may be able to, for instance, say have more amino acids and less serum because they may provide the same nutrients. Um, and some of these may provide multiple functions. Certainly, serum is a good example of that. Maybe supplying both nitrogen and growth factors. And so it's a complex thing to optimize media. All right, so in order to do that, there are traditional ways of doing that, and I've listed some of them here. On the left side, one of the ways of doing that is to use a shotgun approach, basically try a bunch of things and hope that they work. It sounds kind of weird to people in academics that that kind of thing happens, but I know from my experience in industry that was happening more than I would uh, care to think when I joined the company I was with. Probably the next step up in complexity is take one of those 30 components on the last slide and find the optimal concentration of that component, holding all the rest of them constant, and then doing that for each of the 30 components. Not only is that not efficient, but you're unlikely to get a true optimum because you're just disregarding the interactions between those components when you do that. So what are more efficient ways of doing that? Probably the most efficient ways of optimizing media in industry right now have to do with the two I show on the right, factorial, fractional factorial design, or response surface methodology, which collectively I would call design of experiments or statistically designed experiments. Now, this can actually work. Um, it's difficult when you get to levels of 30 components in media to use this because if you think about a factorial design, usually you have a high level and a low level of each of those components, and a full factorial for 30 components would be about a billion experiments, which nobody is going to do. So obviously you can use fractional factorial designs, and there are other designs that we can discuss that would be more efficient than that. Once you reduce the dimensionality, you can use response surface methodology, which is kind of like a experimental gradient optimization method to help you zero in on an optimal media. And this is probably the best approach that's out there now. But again, for high dimensionality, like 30 components and high complexity, um, there's likely to be better ways of doing this. And we started working in this area of research probably about 15 years ago or so, and published several papers in this area about 10 years ago, but focused not on cultivated meat at that point, but on Lactococcus lactis, which is one of the organisms that used to produce cheese, for instance, or yogurt. And I'll show some results in a second, but I want to say first discuss the idea that we worked on, and then I'll show some of the results and some of the results for cultivated meat. So what we decided was lots of times when you're doing these DOE experiments, you actually take an experiment's results and you use that to um, to decide on what your next set of experiments are going to be. But you almost never go back several experiments and look at all that data collectively. So you get information, you use it and you discard it. You get information, you use it, you discard it, and so forth. And we thought, what if there were a way of accumulating knowledge over the course of a process development scheme? And so we did that early on. We're still doing that using a model And we're using radial basis function neural networks and artificial intelligence technique, but there are other ways of modeling to accumulate the knowledge as we're doing process development. And as that model gets better and better as we collect data, we can use that for optimization. Now, at first, the model is not going to be very good. And so if we use that model with an optimization algorithm that finds the optimum in that bad model, it's liable to actually put us in the wrong direction for picking the next set of experiments. And so the method that we developed accounts for that, and it doesn't actually use a very efficient optimization algorithm at first. It purposely uses a less efficient algorithm and then increases the efficiency of that algorithm as we get a increasing knowledge base or better model for the process. So if we think of this box here, this purple box, and you can as a two dimensions. So let's say we have concentration one component on the x-axis, another component on the y-axis, and this is going to end up being a contour plot. So let's say the amount of growth you get comes out of the screen at you. We can do an initial set of experiments, and first we know nothing. As far as we know, any of the combinations of those two components is going to be the same. We then run those experiments, and we can, based on the model that we generate from that, we can pick the next round of experiments. We run the new experiments. We see, is the process still getting better? And if it is, we do a loop. And so I want to actually just show you, this is a work that Zach did, Zach Cosenza in my lab, just to try out his new algorithm. And what you're going to see is the evolving model over time. And this is just a simulation. This isn't real experiments. So you can see this is suggesting experiments down in this lower left-hand corner. But as the model evolves over time, the neural network model, you can see that you start to see the structure and where the experiments are suggested move around until you get to this final model. And this is actually what the model is that we look at. You can then tell that the actual optimum is down here in the lower left-hand corner. All right. So if we had gone back to that beginning and used a very efficient optimization algorithm, we would have picked the wrong optimum. Okay. So I want to now dive into what we're doing in a little more detail now that you have the general idea, and then I'll show you an example of how we used it. So first of all, somewhat simplistically, I want to answer the question first, will this work? Because we know whether it will work or not, because we tried this out with Lactococcus lactis, and we published this about 10 or 11 years ago. So we had a media that had 19 ingredients, and plus we were optimizing for temperature and pH, two of the process considerations. We were doing this at a very small scale. And we tried to do is develop a defined media for Lactococcus lactis because the one that existed didn't work very well. And this is some data from this. What you see here is the maximum optical density at a certain wavelength at 650 nanometers. And what you can see here is this is the defined media that existed at the time. This is the complex media that exists that's common for Lactococcus lactis. You can see it was way better than the defined media. And we actually used this approach that I just showed you. And what we found was a much better growth on the defined media that we developed. All right. So we have way better than the defined media. It's actually even better than the complex media, which was surprising. And what you can see is this column right here is, so I'm sorry, so these are the results. And this column right here at the top is actually the number of experiments it took. And this one where we have this first blue arrow is using statistically designed experiments in DOE and response surface methodology, okay? And it took us about 160 or 170 experiments to get to this media here. The next column And you can see it's about as good in its performance, is using that approach that I just showed you. We're accumulating knowledge. And what we can see is we needed less than half the experiments to get there. We also did a whole bunch of other variants of that algorithm, but I'll just point out this one, which is very much like what I showed. And here we started with more experiments in the initial set of experiments. So it took a few more experiments than this one, but we got an even better media out of that. And so the takeaway here is that this does work, and we found a much better media in half the experiments. But I will say that this methodology was difficult to use. And so when Zach started on this, what we wanted to do is see if we could make it more robust and easier to use. And so this is just, again, a schematic of what Zach is doing. And he has this radial basis function neural network that's accumulating the process knowledge. But he has two optimization methods he's using. One is the one that we used before, the truncated genetic algorithm. And I'm not going to go into the details of that unless people want to know the details of that, and we can discuss it more in the question and answer period. He's also implemented in parallel this DICORS method. That's just another search method that can look through the developing model to suggest what set of experiments to run next. And what he's doing is he's using both, and he's using that to suggest um, the uh, the best set of experiments to run in the next round of experiments. These are some of the details of these two optimization algorithms. I'll just point out that again, especially with the truncated genetic algorithm, uh, what we're looking at is at first we don't have it converge. It could easily convert, a genetic algorithm can easily converge to exactly the optimal point in a model, but we don't want it to do that because we know the models, at least initially, aren't very good um, in the first set of experiments. So as the models get better and better, we let that genetic algorithm converge to the actual optimum, and that's how we find the actual optimum media. These two methods for choosing the next set of experiments are complementary in that the genetic algorithm, truncated genetic algorithm, tends to look at, again, using the same surface, it tends to look more broadly across the whole surface. So it tends to explore areas that haven't been explored yet, which is really important as the model evolves. Whereas DICORS is really good at taking an area that it knows is pretty good already in the model and pinpointing where in that smaller area is exactly the optimal combination of media ingredients. So Zach first tested this on some computational surfaces that are very complex, probably more complex than we'd see with media ingredients, but it's a good way of testing the algorithm because if it works with this, it should work with media. And what he found is, and what the right-hand side is the error, or I'm sorry, how good the process is. So the lower this is, the better the process, and the closer to the optimum. And what you see here is a whole bunch of different surfaces. They all have different names. They're all complex. They have either 10 dimensions or 50 dimensions. And of course, what we're looking at with DMEM is right in the middle of that at 30. And so again, what we wanted to do is bracket what we would see with media. And his algorithm is in the red. And what you see is in every case, for all these cases, Zach's algorithm is at least as good, if not better, than using just, for instance, DICORS or just the truncated genetic algorithm with the neural network alone. And so that's what's making this method more robust, is because we don't know what that surface is going to look like for a new media, a new process. And so by having a method that works on lots of different processes on some arbitrary process, we can actually be more sure that we're going to find the optimal media. All right, so we tried this out with DMEM, just as a proof of concept, DMEM plus FBS. So there's 30 components in the media. We use C2, C12 cells as a model system because it was convenient and being used in Keith Barr's lab anyway. And and they have something like muscle physiology. They will start differentiating. To make this easier, instead of counting cells, we use an Almar Blue metabolic assay. This is Zach's correlation with actual cell count. It correlates pretty well, not perfectly, but pretty well. And what Zach wanted to do is, instead of just looking at, let's say, growth at 2 days, um, he knew that the FBS is the bulk of the cost of that media. And he wanted to also say, what if we want to not only get a lot of growth, but also less expensive media for proof of concept there. So the objective function in the optimization was the growth at 2 days by the Almar Blue method divided by the concentration of FBS as an indication of the cost. So we're really optimizing in this process is this objective function that you see in the lower right-hand corner. So here are the results, the initial results. And what you see here is, I should say, to compare, we did use a fairly efficient design of experiments, fractional factorial experiments followed by response surface methodology after reduce the dimensionality. And this is the data for our method, the different rounds of 10 experiments at a time, starting with initial 30 experiments and then 10 experiments at a time. And what you see is that if we look at just the growth, we can see that our method, which is in the red, and the DOE method, which is in the blue, gave significantly higher growth than DMEM, which is in the yellow, after 2 days. And so it worked fairly well. And if we have not only the growth, but the growth divided by the cost of FBS, we actually see that response surface methodology and DOE gave us this blue line. The best methods through the best of media that we came up through our approaches in the red, and DMEM is down here. And so we can see is that we get a very similar process using our method and experimental design. But the key difference here is it took us 30 to 40% fewer experiments to do that. And for those of you who are doing these kind of experiments every day, you know that these are very difficult experiments, very time consuming and very costly. So reducing the amount of time you need for process development by 30 to 40% is huge. And this corresponds with what we saw with Lactococcus lactis, where we saw a 50% decrease in experimentation. So we can look at that and try and understand now what's different about these different media. And I don't want to spend a lot of time on that because I want to leave time for questions, and I'm running over. But maybe what I'll do is talk a little bit here about these. What we show on the y-axis here is for each of the ingredients in DMEM, which for our method, which concentrations we tried. And zero does not necessarily mean zero concentration. These are scaled. There was some base concentration of each of these components and some maximum. So 0 corresponds to the minimum, 1 to the maximum. And what we see here is that the level chosen by our method is in red. The level chosen by the DOE method is in green, and the level in black is in DMEM. And I'll just point out here, we're choosing lower levels of FBS, and we're forcing that to happen because we're penalizing for having high concentrations. Just as another example, glucose will be higher in these new media than they would be in the DMEM plus serum. All right, so some of the differences for DOE, FBS is low, we're forcing that to happen. Glucose is higher middle, and you can see some of the other things that we found. For our method, FBS is also low. Glucose is in the middle, where glutamine, for instance, is at higher levels, and salts are higher, and so forth. This is an important slide that I want to just point to and say that what we showed was at 2 days, our media gave much higher growth. When we then used them and passaged out 5 passages, so probably another 10 to 12 doublings, what you can see is the DMEM works better. So at first, what we optimized on is here, and our media are better. Out 5 passages, DMEM is considerably better. So that's really important. It means that you have to pick the right objective function. The optimization algorithm actually works, but you need to pick the right objective function to use it effectively. And I will say, one of the things with black box models is that, they can work very effectively for optimization, and this is kind of a black box approach. It doesn't do as well in terms of if you run into a problem later. Let's say you get variability in a media ingredient, it doesn't do as well later in trying to understand and fix that problem. And that's why I think it's really important when you're doing this to, in parallel, do the opposite of black box model. It's called a white box model, where you're very carefully trying to understand the physiology in the cells. And that's something that we're trying to do now, trying to understand how are each of the nutrients used in that fermentation process. And this can help go hand in hand with a black box approach. And I'll just to point out that, eventually the media ingredients are probably going to become more complex as they become less expensive. We just published a review recently in Comprehensive Reviews in Food Science and Food Safety, where we go into this in a lot of detail. But suffice it to say here that the complex ingredients that are out there could easily supply almost all the nutrients needed for animal cell culture, and so it's an area to look in um, as you're moving forward. And this is just the paper that's out as of 2021 and online. So I want to summarize. Apologize for running a little bit over, but to summarize, media optimization is really difficult. Um, that's why advanced experimental optimizations like the one that we're developing are really important and are going to be way more efficient than traditional design of experiments. But you can't do that in a vacuum. You should also be understanding your system, because that's going to help you in the future, not only to help your black box approach work better, but also for troubleshooting your process as it goes into production. So with that, again, I just want to acknowledge the people who did this work and the funding for this work. And at this point, I think I'll stop sharing and open it up for questions.
Fantastic. Thank you so much, Dave. That was huge. I think the way you illuminated the use of computational models to help us be a lot more efficient and precise with our media optimization. I do think like contrasting the efficiency of computational models to the really expensive kind of iterative experimentation that you see with traditional methods. It's really exciting, and I'm excited to see if this helps spark or inspire additional multidisciplinary approaches to media optimization.
And I will say, that the problem that the cultivated meat industry is facing with media is not unique. It certainly is true of any fermentation industry, that this is a very similar problem.
Mhm.
And I think there's just maybe less expertise and more cost. Cost is driving it more. Um, so yeah, I mean, I think the reason that we started developing these techniques 15 years ago is because uh, we these are tools that I would have loved to have had in industry and didn't have them.
Mhm. Yeah, it's so fantastic that you're developing these tools for the cultivated meat field now. We will move on to some of the questions that have come from the audience today. Those of you who haven't yet added your questions in, please do feel free to drop them in the Q&A section down in the Zoom bar. And for those of you who are asking, yes, the seminar recording will be uploaded to our YouTube channel, and you'll be able to watch Dave's presentation back again at your leisure. So one of the questions from this chat is whether or not you plan to make these models that you've been developing open access or open source for the cultivated meat field.
Yeah, so that's the goal. And I would say when we first developed these models, this approach 10 years ago, we published it. This is all out there. But having said that, if you're not an expert in neural networks and optimization algorithms, it's not going to be easy to use. The goal for us is, for some of you who are interested in, let's say, design of experiments, there's all kinds of possibilities out there. But one of the programs that we've been using for years is called Design Ease, um, uh, by a design expert by StatEase, and it's basically a program that's very, very easy to use for designing statistically designed experiments and will help analyze them. The goal for us is to have something like that. And certainly, we published. I didn't point this out, but Zach published his algorithm. I think last fall. We're about to publish the work that I just showed. He's waiting for me to read it. He told me I have to read the paper by tomorrow. Um, but whether we'll so the methods are already out there. You could conceivably do them yourself, use them, develop them yourself. But the goal for us would be to make them robust enough so that they can be basically canned software that you take off the shelf and just use it as a tool when you're doing your process development.
Very, very cool. I can see that paying huge dividends for the cultivated meat field. And it's so exciting that you're doing this research that will be a force multiplier for all of the research efforts that follow.
Mhm.
Very awesome. I'm going to dive into a few questions about um, kind of lowering the complexity of cell culture media.
Mhm.
So Aditya asks, could you potentially use engineered microbial lysates to bring down the complexity of the cell culture media? Since most of the ingredients are probably produced through fermentation anyway, could you perhaps engineer a microbe with just the right proportions to use directly?
Well, I think the short answer to that is technically yes, you could do that, but I'm not sure. I mean, it could be an interesting approach, but I'm not sure that it will end up being as inexpensive as other approaches. So I showed that table right at the end very quickly. All of those columns were agricultural byproducts. So there were things like corn steep solids, corn steep liquor, soy flour, molasses, and things like that. These are, to some degree, byproducts of agricultural processing that's already happening. And if the nutrient um, composition of those ingredients work for cultivated meat, that's probably a less expensive approach because that processing is already being done, and you don't have to create a new process. If you have to create a new process with a new bacteria, for instance, or maybe a yeast as an example, it's probably going to be, at least initially, very expensive until it gets to such large scale and cultivate meat get to such a large scale that the price comes down. Um, Also, I think what you want to do is make sure that anything that you're doing is food grade. All these agricultural residues are all food grade, as would be using something like a yeast extract, which is another possibility. Yeast extract is used widely in fermentation media, and the Saccharomyces cerevisiae is obviously food grade, and so that might be just a twist on your suggestion that would make it easier to implement because that's already being done at a large scale and being used as a fermentation media ingredient and as a food ingredient, as a flavoring.
Yeah, thank you. I do think it's really insightful that, you know, um, using cultivated meat production as an opportunity to create kind of a circular economy, lowering the cost of cell culture media by using existing agricultural waste streams. I think that makes a lot of sense. And maybe Aditya's question was specifically about engineering those growth factors.
Mhm.
But yeah,
So I will say, that one of the things that you want to avoid, and this is from my experience in industry, is having to create two manufacturing facilities and two processes to make one product, because that's going to be really expensive. And whether it's to create the nitrogen and carbon and vitamins and mineral kind of background, or specifically growth factors. Um, if you think about that, to make a recombinant growth factor, you or someone else is going to have to produce a whole nother manufacturing facility and a whole nother process to make your cultivated meat. And that's why, if you can avoid that, it's going to be better. And it may or may not be possible. I mean, that's certainly a key issue is, can you find a way to replace those growth factors with something that's coming from a product that's already out there being produced large scale inexpensively?
Yeah, thank you, Dave. All right, pivoting slightly to ask a question here about uh, whether there are any concerns that the cells will eventually enter like cell death with limited FBS concentrations, even if the low serum media is being replaced super regularly and recycled.
Well, I think that's going to depend a little bit on the cell line, and assuming you have a cell line that can um, grow forever on FBS, I think it's a matter of finding the right media to have them grow forever on a media without FPS. I think if you want to think about this optimistically, which I like to do, I think it's a matter of finding the right mix of ingredients that will allow that to happen using optimization tools like the one we talked about. The If you want to think about this pessimistically, I think it still happen because eventually someone's going to take, if they haven't done it already, serum and completely characterize every single molecule in serum to find out which are the important ones to include in cultivated meat media. I mean, that's the worst case scenario. I mean, it might happen academically. I don't know that it's going to need to happen. Someone will probably do that eventually. Um, yeah. So I'm optimistic that having the right media with a cell line that's going to grow 50 generations or whatever it needs to grow is doable.
Awesome, thank you. Here's a question from Mariana. When optimizing the media for cultivated meat production, is the kind of object of interest or the objective to maximize the growth in biomass, or is this some other output that you're measuring? Um, I assume this, yeah,
Yeah,
it depends. Go ahead.
I mean, picking the right optimization uh, objective is really important, and I think there's going to be many objectives that cultivated meat companies will have. I think the first one is to grow the most cells. The second one is cost, and those may be equally important. But growing the most cells is going, and the fastest and the most cells is going to be important because that's going to determine the capital outlay for the equipment. Um, if you go to a low cell concentration, your equipment is going to have to be much larger, right? So you want to have process intensification, is what chemical engineers call that. Cost is going to be important, but then after cost and um, and cell mass, this is after all a food, right? And so we're going to have to optimize for nutritional value, right? And it's likely the nutritional value of the cells coming out is going to change with the media. And because again, it's a food, we're going to have to optimize um, for flavor, right? And so the sensory properties of that product are going to be really important, and they're almost certainly going to be affected, maybe positively, by the media ingredients. And uh, so that's another optimization that has to be done. And then there's the differentiation, right? And then because it's a food product, food safety is going to be important. So we're going to have to be really careful about using raw materials that are going to be easy to fit into a food safety regime.
Yeah, thank you. A related question from Reza at Virginia Tech is whether or not through your experiments you measured the condition of the cells outside of overall growth.
So, so I will say we're starting to do that more. So, uh, I think eventually looking at simple cell viability is going to be important. It's all, because viability is going to be important during scale up. It may or may not be important in the final product, but it'll be important during scale up. Certainly, as we get to differentiation, either wanting it or avoiding it, that's going to be important. That's going to be important more in process development than manufacturing. I think one of the things that, and we're going to have to worry about that to make sure that cells grow as long as we want them to grow and differentiate when we want them to differentiate. I think it's certainly true in manufacturing that you want to be in a place where you're under control of your process, but that you're measuring as little as possible. Um, and You only want to measure the very, very key things, because as you go along, and this is going to depend on the regulatory environment that develops, um, any things that you measure that fall under regulation are going to make your life more complicated. So you only want to measure the things that are really key. But I mean, certainly starting up in where we are now, understanding the state of the cells in terms of their differentiation state and their viability and so forth is going to be very important.
Thank you. There's a question here from an attendee about how you might expect to account for variations in lot to lot and vendor to vendor inconsistencies of commercially available defined media, knowing that that's typically a pretty big problem when we're talking about commercial scale production.
Yeah. Yeah, that's huge. I mean, it's not unusual for other fermentation processes to have plus or minus 20% performance, which is way larger than you want to be in manufacturing, but it's not unusual. Um, I will say that, whether it's a defined media or it's using these agricultural waste products, there's going to be variability. Um, there are a couple ways of dealing with that commercially. One way is a way that I've heard of people doing this. And let's say you're using, I'm going to pick soy flour as an example. Soy flour will change based on where the soy was grown and the climate that year, the weather that year, if it's a wet year or dry year or whatever. And what some large producers, fermentation producers, will do is if they find, let's say, a soy flour that works really well, they corner the market on soy flour for that year to hedge their bets that next year's soy flour is not going to be as good. So on a large scale, you could do that, assuming you have the buying power to do that. The other thing is to try and identify which of those which product's variability is the most important in the outcome of your fermentation, and then to take that, basically, where all you have is a lot number and you can't optimize on a lot number, um, and make and do a detailed analysis of that one ingredient to figure out what's causing the variability, and then create a spec based on that uh, couple things that are creating the variability, and work with your supplier on those things, um, or find a new supplier with less variability. And I think as the industry grows, that's going to be more and more possible. A good example is, let's say, lots of people use peptone in a lab to grow bacteria or yeast or whatever, right? Um, if you're using Diffco, just as an example, that's a lab grade reagent. There's a lot less variability there than in the much less expensive ones that you might buy, um, from other vendors. And they know that but they also know that cost is important. And so, you may have to balance between variability and cost.
Fantastic. Thank you, Dave. There's a question here from Nara, which is about how extensible the algorithms that you're building and the models that you're building are across different cell types and meat relevant species, and perhaps also across different structured or unsophisticated cuts of meat. Do you have any insight there?
Yeah. So the algorithm is really agnostic to what it's optimizing. So it should not matter. I mean, we're developing this for fermentation media because that's what I work on, but you could use this for, you know, I don't know, processes that have nothing to do with food at all. Like let's say you're producing, I don't know, microchips in the Silicon Valley, and there's a whole bunch of different process inputs that go into that you need to optimize to get, high quality or high throughput or whatever. It's it's just like that. So, just like the algorithm, uh, design of experiments could be used for anything. It doesn't have to do with, a specific type of media or whatever. What you won't use is the models that we develop for our own processes. The model will develop itself. Let's say you have your own media, you'll do an initial set of, let's say, 30 experiments, put that data into the algorithm. It's going to start generating model for your own process. So you won't be using a model that we generate. You'll just be using the algorithm that we generate.
Awesome. Thank you so much. There's a question here, a suite of questions here around the kind of task of removing FBS from the cell culture media that will be involved in cultivated meat production. And people are generally curious if you can speak to that, what you think some of the approaches might be to removing FBS from cell culture media, whether things like algal extracts or mycelial extracts or hydrolysates might be used as FBS replacements in media. And yeah, just your overarching impressions of the trajectory of removing FBS from the dominant cell culture media solution.
Yeah, and I think that's obviously the ultimate goal for everybody, and certainly in terms of cost, that's the main cost driving factor. Um, yeah, we're starting to work on that now in our lab. Now that we have proof of concept that the algorithm works, we're starting to work on, can we use it to choose the best replacements for serum? And I think the simplest approach, but not necessarily the best approach, is to use um, defined growth factors. So probably produced recombinantly, um, and uh, and that's the simplest approach, because I think a lot of the other ingredients or a lot of other components of serum probably can be replaced with some of the things that media kind of ingredients that I showed on my slides. But it's the growth factors that are going to be much more important. So GFI is funded a project at University of Toronto, for instance. Peter was working on uh, recombinant growth factors that he feels could be produced inexpensively, and maybe much more active than the ones that are on the market, which I think is really cool. So that's one approach. The downside to that, as I mentioned before, is again, it means that you need multiple processes and manufacturing facilities to produce your one product. And so if you could replace it with one of the other options that you mentioned, Amy, the person asking the question mentioned, um, that would certainly be better. And I'm sure people are working on that. We're working on that as well. I think you obviously want a molecule that's going to do what the growth factor does, uh, and but do it with a much less expensive molecule. So.
Thanks, Dave. I dropped a link for those of you who are interested to Peter Sturgios's project at the University of Toronto there in the chat, in case you'd like to check it out. There's a question around whether you have either an intuition or know of any forecasting projects being done to anticipate when the media costs for cultivated meat might be reaching price parity or cost parity with traditional meat.
Actually, I'm guessing there are people at GFI who could answer that more easily than me. Um, so,
Yeah.
Peter's here. Good. Um, So I don't have that estimate. I would personally, if I were looking for that answer, I'd probably go to GFI's website and look at some of the stuff that Liz and Elliot have written, because I'm guessing um, you'd find it there. I mean, one of the problems is that right now, companies that are producing, let's say, growth factors themselves recombinantly, um, I don't know that any of those companies are huge companies and probably don't have enough to handle really large scale production of cultivated meat. So, if you're thinking about, they may be able to contract manufacture those kind of products, and they probably are. But if you're really thinking about supplying the industry, you're probably talking about building new facilities, and that's it's going to take, by the time they're designed and built and so forth, it's going to take at least a couple of years, and that's if you started now, right? So um, I think there's going to be some transition where the media and the product are going to be somewhat more expensive, um, until either you have large scale suppliers of things like growth factors, or you figure out how to get rid of the growth factors. And if you can figure out how to get rid of the growth factors completely, then it's a much shorter time scale, I think.
Yeah, definitely share your intuition there. In terms of GFI resources, Liz did write a really fantastic paper examining different scenarios for bringing the cost of cell culture media down. Maybe my colleague Renee there in the audience can help dig up the link to that for those of you who are interested. And also GFI has been conducting our own LCA and TEA for cultivated meat production, led by senior scientist Elliot Swartz. That will be released later this month, so you can stay tuned to also dig into what our projections for reaching price parity might be.
And I should mention, I mentioned the review on media that we published just recently. You take a look at that. It doesn't really talk about timelines there, like some of the material that you're talking about. Ned Spang's group in food science at UC Davis also just published a TEA that I think points to media as probably the most important factor. And so that's another place to look as well. So that's Edward Spang is the corresponding author on that.
Awesome, thank you. Here's a question from Sarah Oliveira, who's one of your fellow grantees. Um, do you know of any cell-related behavior that the model did not capture, um, that kind of explains why it worked well for the first passage, but not the later passages? Sarah's working on biomodels for 3D bioprinting and using DOE at the moment.
Right.
Besides having multiple variables, those variables also just come from different sources and not just the media formulation.
Yeah. Well, first of all, I'd be happy to talk to her about some of the stuff we're doing, maybe collaborating. But I think you know one of the one of the really good things about the kind of approach that we're taking is it can be very fast, and it doesn't require you to take the time to really understand the system. The model is a black box model. It's like drawing, on a simple scale. If you had, one parameter, it's like linear regression, right? You're just doing finding a correlation between the inputs and outputs, and so there's not really any real true process understanding that's going to come from that. There's some, because you can get an idea of from how the algorithm pushes uh, the area that it's looking in for optimization. You can get some idea of which parameters are most important and so forth. But it's not really understanding the physiology, and that's why I think it's important in parallel. My experience has been in industry and academia. It's always important to try and understand what's going on a fundamental level at the same time, and that's what we started doing, and that's what Ted O'Neill, my lab, is working on, and trying to really understand, when are certain nutrients used, um, when are other nutrients not used, and so forth, and you know why it works for a while and not after like multiple passages. I don't know. It could be that the cells coming in, um, which where, if the inoculum is prepared with serum, maybe there's enough growth factor carryover that in the first growth that you don't have to worry about it. And yet that maybe gets diluted out if it gets, let's say, attached to the surface of the cell or internalized into the cells. Maybe that gets diluted out as you get cell growth, and so maybe it's something like that. But we don't know yet for sure.
Awesome, thank you. We have 24 questions left and
No,
only
no,
5 minutes left on the clock. So perhaps those of you who don't get your answers or questions answered today can shoot Dr. Vlach an email and take things offline.
sure.
But I think it would be fantastic to get your perspective on this last question here from Zihong Wang from the University of Tennessee in Knoxville. Um, you have a really interesting background, kind of an unconventional background coming from viticulture and the wine industry and then entering cultivated meat. So the wine industry seems pretty different from the cultivated meat industry. And Zihong is wondering what made you decide to step into this field? And is there something that was kind of overlapping between these two fields, besides the fact that fermentation is an aspect of both,
Sure.
um, and there are kind of cross-platform similarities. Yeah, is there something that you found helpful from your previous experience that's that you've carried over to cultivated meat?
Yeah. Well, I would say, uh, in some ways, the specific experience that I'm applying to cultivated meat came from working on fermentations other than wine. And so I've done a lot of work on pharmaceutical fermentations, and both in industry, and then when I joined Davis, working on recombinant protein production in various organisms and so forth. Um, and there, the media optimization problems are very, very similar. Um, I'd say what the wine background does for me, and is especially being chair of a wine department, is that I interact with industry all the time, and it's basically a food industry, and B, it's heavily rooted in agriculture. And I think those are two really important points here, because when we're dealing with cultivated meat, you're dealing with, first of all, a food product, and second of all, if you're not, you're going to have to be dealing directly with agriculture, either A, because you're going to use raw materials that are coming from agricultural waste streams or byproduct streams, or because you're going to be constantly dealing with the conventional animal agriculture wing of meat production, and um, through regulation, through production, through distribution channels, and so forth. And so having a really good understanding of what's going to fly with a farmer, or what's going to fly in food production, is really the connecting point between my experience with fermentation optimization and creating a food product like cultivated meat. That's a good question. Haven't really thought about it that way before.
Thank you so much. I guess the very last question that I'll ask here, there are a few sprinklings of activity in the chat. Wondering what your how you might be looking to collaborate with other stakeholders in the field, or your lab might be interested in collaborating with other stakeholders in the field, whether on the academic front or the industry front. Would you like to speak to any of the collaborations you're presently seeking or would be open to exploring?
Sure. Yeah. So I mean, we're always open to exploring collaborations, and I would speak for all the other team members here that I'm sure that they're open to collaborations, and we are actually starting to have collaborations not only at UC Davis but outside of UC Davis, quite broadly around the world. Um, I'm very interested in that. I work with in my job with the wine industry, I work with companies all the time doing joint research projects, and academics around the world on joint research projects. So very open to that. And if people are interested, they should just contact me. Um, I think that's going to be really important. Davis is somewhat unique in that it's a large group, large um, diverse group of academics working on cultivated meat. And, I think up to this point, there's been, I would say, uh, more individuals here and there who have worked on cultivated meat in academia. Um, and uh, so I think, yeah, I'm very open to that. And I think it's going to benefit the whole field to have those collaborations going. And I've spent quite a bit of time, I guess it's a little more difficult in a pandemic, but quite a bit of time going around and talking to startup companies. Uh, and it's always interesting to do that. They all come with different perspectives and expertise and experience levels and understanding of the food environment they're entering and so forth. So I'm very happy to do that as well. I'm very used to working with companies and talking to companies, and I enjoy that part.
Awesome. Well, the chat is just flowing with high praise and enthusiasm about your research and the broader efforts you have underway at UC Davis. I'm really excited to continue following the progress of these research projects and of the ecosystem that you're building at UC Davis. Before we wrap up today, are there any final thoughts that you would love to leave our community of scientists and engineers with?
Just that, this is a hugely exciting area. It's very rare that you get to, in a scientific career, to enter a field that's just starting. And it's a really unique experience, and it's exciting and it's fun. And, you know, I guess savor that. I guess you can say that because it's a food product, but savor that because um, you don't get to do this very often in your career. And so I'm excited to be a part of it, and excited to be working with GFI and all my collaborators here at Davis. And it's exciting to go to the grantees meetings and hear what's going on in all these other labs around the world. Um, it's just a lot of fun, and so I'm happy to be part of this.
Awesome. Yeah, echo those sentiments 100 times over. It's so rare to be kind of at the cusp of creating a new paradigm for
Mhm.
feeding the world, and a really exciting time to be involved with this industry because we are so early stages that every player plays such an important role in defining the trajectory of our food system. So Dave, this was a huge pleasure. Thank you so much for sharing your knowledge with us today. Thank you, everyone in the audience, for tuning in from every corner of the world. It's always so encouraging and inspiring to see just how global this community is. Next month, we will be joined by another GFI grantee, Dr. Marianne Ellis, to learn more about some recent research advancements she's been driving forward. Thanks again for everyone. To those of you who attended and are part of our community, we will see you again next month. And for those of you who are not yet part of our community, you can sign up at gfi.org/community. It was a great pleasure to see you again today, Dave, and we will see you all again soon. Thanks again. Bye.